just, oh, thank you. Recording started. Garrett has just joined the Texas Extension Service, at Texas A&M College Station. He has most recently been as a postdoc fellow at the USDA Baton Rouge, Louisiana laboratory. He has an unusual background. He worked for a number of years in North Dakota for a large commercial beekeeper. Got his master's at North Dakota State University, and then he said in nutrition and queen quality, and then his PhD from Purdue in 2022 before going to the Bee Lab in Baton Rouge. His presentation tonight, importance of drones for quality queens. So I'll turn the program over to Garett Slater. And welcome. Thank you for joining us. I will indicate that we had Garrett set for last month, but he had a rather significant event occur. I'll let him introduce that. And I appreciate his willingness to come and be with us this month.
Well, thank you, Dewey, and thank you, the panelists. I'm really excited to be here, and like I said, I was originally scheduled to be here last month, but we just recently had a baby. So Dewey, he said you wanted me to add a picture, so here's a picture of my little baby girl, Sage. So she's healthy, the mother's healthy, and she's born on the 23rd, the exact day I was supposed to present. So I'm really thankful you guys are willing to accommodate and make that work. So I'm getting plenty of sleep. I'm not sure about the mother, but I think we're all doing well. So thank you for working well with me. So yeah, so before we begin, so first I'm the new honeybee extension specialist for the state of Texas. Surprisingly, we haven't had a honeybee extension program beforehand, so I'll be sort of spearheading those efforts. I'm not sure if Texas is part of WAS, but I'm sure there's probably some Texas beekeepers, especially the western part here. So, here's sort of an introduction to myself. So today I'll be talking about drones. And I started doing my research on drones during my PhD at Purdue University. But I've been interested in drones really my entire life. And it really started during my work with a commercial beekeeper. Beekeepers reporting that queens were failing at a high rate. They're purchasing queens and they're failing within 6 months, where they should be reproducing and thriving for 2 to 4 years. And so I've always been interested in this queen quality, queen failure issue. And one thing I was always surprised in is, what about the drones, right? We always focus on the queens and the aspects of queen failure, but we sort of forget about the drones. And really, when I started doing my research on queen failure and into my PhD, there was so little research on drones, even though they're just vitally important to not only the colony genetics, but also the queen quality metrics. And so really today I'll be focusing my drones, some of my research during my PhD, and I'll probably touch on some of the research I'll be doing here at Texas A&M University. So before we begin, I'd like to kind of give a summary about the castes and the sexes within a colony. And so a colony typically has somewhere between 5 to 10,000 individuals during a given year. And at the top, we have the queen. The queen is a female and the primary reproductive of the colony. She's also diploid. And so the queen's only role is to really reproduce and lay eggs and really sustain colony size and colony growth. At the bottom, we have workers. Workers are also diploid females, and they're typically not reproductive, and they perform a wide range of tasks within the colonies, ranging from foraging to nursing to garden behavior. But then we have these drones right in the middle. Now, these drones are, about the same size as queens, a little bit larger, but they have these massive abdomens, which is packed with their reproductive tissues for sperm production. They have these thoraxes that have a lot of muscles that allow them to fly to drone congregation areas and other mating areas. And then they have these huge eyes that allow them to navigate through the air. And at a given time, there could be upwards 10% of the 20% of the colony members of just drones. And so that can be, a few hundred drones within a colony. And so it really depends on the area you are, but your colonies are probably beginning to produce drones right now, and they're probably beginning to go on these multiple mating flights. And so when we think about colony health, we really focus on queens and workers, but we do forget about drones, even though they're vitally important to colony health. And we sort of view them as, because they really don't perform any tasks within the colony. Their only role is to mate. but this mating is really important for everything we look at as beekeepers. And so, like I mentioned earlier, when we survey beekeepers and we ask them why do your colonies die, usually it's viruses and Varroa number one and two, but queen failure is usually number two or three on the list. And so this is a major issue that faces beekeepers. And I'm not sure if you have David Harpy speaking to your group beforehand, but he talks a lot about these surveys. And one thing he sees is queens are or beekeepers are reporting that queens are failing at a high rate. And so, one of my questions was, why are queens failing within 6 months when, I talked to beekeepers from 20 years ago and, from their anecdotal evidence, they're surviving for 2 to 4 years. What is that change? And we've seen a lot of research focused on the queen aspects of failure, right? Like Alison McAfee's done some really great work. Julian Rangel at A&M's done some really great work. But, one thing I noticed was we're just focusing on queens, but really, what about the drones? Is there some issue with the drones that the queen's mating with? And maybe that's a reason that queens are failing. Because if queens are mating with drones that are low quality or producing low quality sperm, that can have a massive effect on the queen because she's storing that sperm and using that throughout her lifespan. But if she's mating with drones that are producing sperm that's, let's say, 50% dead, that queen's more likely to fail earlier on. And so, one thing I wanted to do during my PhD was really focus on drones, understand the biology, and see are drones a major driver of queen failure? And so when you think about do drones contribute to queen failure, there's really two major aspects. Or when we think about are drones important for colony health, there's two major aspects. One is they can contribute directly to queen failure. So this is a really great study done by Dr. Jeffrey Pettis, and at the time he was at the Beltsville Bee Lab, USDA Bee Lab in Maryland, and he was hearing these reports from beekeepers. And so he did this really simple but really powerful study. He sampled queens that were failing in the East Coast, queens that were failing on the West Coast, and he compared them to the control queens at the USDA facility in Beltsville. And what he did was he pulled up the spermatheca, which is the sperm storage organ, and he measured what percentage of the sperm was alive versus dead. Because what we'd expect for a high quality queen, we'd expect that sperm to be nearly 100% alive, at least 90% viable or alive. But what we found is for the control, that was true. 90% of the sperm for those queens were alive. But for the queens that were reported failing in the East Coast and the West Coast, they were only storing sperm that was 50 to 60% alive. And so if we look at these numbers, I mean, 50% of the sperm that the queen is actually storing from these failing queens were dead. And so one reason that, these queens could be failing is because, there's mating with poor quality drones that are producing poor quality sperm. Now, this could be a queen issue, right? It could be the queen's ability to store the sperm, or something happened to the queen that indirectly affects the sperm. But this could be due to the drones that the queen's actually mating with. Second, when we think about why drones are important to colony health, we think about colony genetics and colony success. And so when you think about just any trait you're interested in as a beekeeper, whether that's mite resistance, virus resistance, productivity, different landscapes such as Texas versus, let's say, Oregon, or colony level performance, 50% of the genetics that contribute to these traits are due to the drones, right? So 50% from the queens, and the other 50% due to the 15 to 20 drones that the queen's made with. And so, what I tell beekeepers is, drones aren't only important for queen success and potentially queen failure, but also all these different traits that beekeepers are interested in. And so, at Baton Rouge Bee Lab, for example, we've been breeding for pole line, which is a VSH selected stock, and we've also been selecting for higher productivity and pollination services. And we see that, a lot of the genetics are also due to the drones. We are starting to breed for virus resistance at the Baton Rouge Bee Lab. We see that drone genetics are contributing to this, and also productivity and colony performance. And so, just take a look and just think about which traits you are interested in as beekeepers. 50% of this is probably due to the drones. And so, I want you really the biggest takeaway here is that, drones aren't just these, mumbling males in the colony. They're really important for a lot of the different traits, you are interested in as a beekeeper. And so when I started my PhD, one of my major questions was, what actually makes a successful drone? And, I did a full literature review right when I started my PhD, and there's so little research done on drones, even the basic biology of drones. And so what I did was I sort of wrote just a review. It's open access, and you can probably find it in Google Scholar, or I'm happy to send it after this presentation. But I basically just made a literature review about, the biology of drones, the genetics of drones, and really what are the important traits that make a high quality or successful drone. And from this literature review and some of the other research I did, I found there's really three major points that make or break a drone. One is sperm production during development. So, honeybees are whole metabolists. So they go from the egg stage to a larval stage, then a cocoon spinning stage into this pupa stage. And this stage takes about 24 days. Now, for a honeybee male drone, this is extremely important because this is the only stage of development where drones can actually produce sperm. After they pass this developmental point, when they become adult males, they can never produce sperm ever again. And so, if anything happens during the stage, it can cause irreparable damage to the drones and really produce sterile or infertile drones altogether. And this is really unique to honeybees. Even if you look at other taxa or even closely related bees like bumblebees, they can produce sperm continuously throughout their lifespan. But for honeybee drones, it's really unique and it's really restricted to this developmental stage. And so honeybee drones take 24 days to develop. And then once they emerge as adults around 24 days, they spend what I like to call maybe the teenage stage in the middle of the brood box. And I like to call it the teenage stage because they're really dependent on the workers for everything. I've actually taken young new drones and put them in an incubator, and most of them die because they're really dependent on the workers for the food, for the warmth, and really everything. But this stage is really important because this allows the drones to actually reach sex maturity. And I'll talk about this a little bit more, but the sperm actually has to migrate from the testes, where the sperm is produced, to a separate organ called the seminal vesicle. And once the sperm is in the seminal vesicle, that sperm can actually be inseminated into the queen, because the sperm can't be inseminated to the queen if it's in the testes. It actually has to be in this separate organ called the seminal vesicle. And so that's why this sexual maturity stage is really important for the drone, because it allows that sperm to migrate and be available during mating. Now, once drones reach sexual maturity and become mature drones, they go on these multiple mating flights, and they go to these areas known as drone congregation areas. And if you guys have seen any or did some UAV drone trapping of them, please let me know, because I'm really curious about why drones find these mating areas known as DCAs. But what drones will do is they'll kind of go on these orientation mating flights, and they'll go to multiple DCAs. And then once they get older and they know, okay, I have to mate or I'll die and not be successful, then they kind of pinpoint one DCA and go back and forth behind it. And so I really think the mating behavior is really fascinating, not only how drones find these congregation areas, but why are they choosing these areas? And once they're in these areas, what they do is they sort of form these clusters, and they wait for a queen to arrive. If a queen arrives, they form these massive drone comets, chase after the queen, and the queen's only going to mate with a few of these males. And at times, these DCAs can have upwards to thousands of drones at a time, and so it's an extremely competitive environment. And there's some really cool videos showing the drones kind of running into each other, knocking each other out of the way to really compete for the queen. Now, once one of these lucky drones mates with the queen, their whole, I guess, reproductive tissues are basically pulled out of them, and the drones die. So they can only mate once. They can never mate ever again. And so I really think the mating system for honeybees is really fascinating and really cool. But really the biggest takeaway here is when you think about what makes a successful drone, it's really split into these three major points. One is sperm production during development, their ability to reach sex maturity during the first 10 to 14 days of their adulthood, and then their success in finding a queen and also successfully mating with a queen. And if anything happens at one of these 3 points, that can cause the drone to not be successful, maybe become fertile, and not successfully mate with the queen. And so I wrote about this a little bit in my review. And one thing I wanted to do was sort of address what's causing poor drones, and is something happening at one of these three major stages. And so I'll sort of give a little overview for for each point because I know this is probably a lot of new information for a lot of beekeepers and people. And so, like I said, honeybees are holometabolists. That means they go through complete metamorphosis. And what that means is they go from a larval or a grub stage all the way to a pupa stage. So basically transforming their tissues, and they're going from one stage, like this grub stage, all the way to a pupa stage. It's very, very similar to a butterfly, for example. Now, workers and drones, they all go through about 5 to 6 days of this larval stage. And, if you have experience looking at these larvae, as you come back the next day, they get bigger and bigger and bigger. And we like to call them different instars because they're kind of, sort of in a way molting to a different size. Now, once these drones reach what we like to call a fifth instar, this is when the drone larvae are really big and almost seems like they're almost overflowing out of the cell. And you actually might have some of these in your colonies at this point, and maybe take a look. But if you see these drone larvae that are really large and they're not quite capped yet, but they almost look like they're so big that they don't fit inside the cell, this is probably a fifth instar larva. And this stage for a drone is really important because this is the first stage of sperm production, and we call this spermatogenesis number one. But it's really a fancy way of saying this is when drones actually begin producing sperm. Now, once the cell is capped and the larvae become upright into what we like to call a pre-pupa stage, this is also when the drone begins spinning cocoon. You can't see the cocoon, but if you have a microscope, you can actually see them spinning a cocoon. But this is actually the second stage of sperm production called spermatogenesis number two. And so you think about, when does a drone actually produce sperm? It's only during this 2 to 3 day window during the fifth instar late stage into the pupa stage. And after that, drones can never produce sperm ever again. And so, this 2 to 3 day window is extremely important because if anything happens during this stage, this can deplete sperm production and really cause a drone to be infertile. Now, once they reach what we like to call a purple-eyed pupa stage, once they enter the pupa stage, their eyes become melanizing from sort of a white color all the way to this kind of dark purplish color. When they reach this purple color, this is when the sperm tails are actually formed for the drones. Now, so I just so much to do a thought experiment for you guys, just to think about what could happen to the stage that could cause drones to be sterile or infertile. Now, what happens? One is, drones are mite magnets, and so if you've ever put in a drone comb into a colony or opened up a drone cell, there's usually a ton of mites feeding on the fat body during this stage, especially this pre-pupa stage. And so, for example, if your mites are really infested, the mites could be feeding on the drone pupa and really causing that drone to be sterile or infertile. And so mites could be a big issue. Another thing is, miticides. And so just kind of think about, the stage is really important. And if anything happens during the stage, this can really impact drones. Point number two is sexual maturity. And so after 24 days of development, drones will sort of hatch out of their cell and eclose into adult drones. Now, once they emerge as adult drones, what they'll typically do is go to the middle of the brood box. And so let's say you have, double deep brood nest, they'll typically be where the young brood is and the young brood's emerging. And they do that because they want to be around, the young nurse bees. And they want to be around the young nurse bees because the nurse bees are really taking care of the drones. And I sort of think that's why drones get such a bad rap, because they're so dependent on the workers for everything, and they really can't do anything for themselves. But this is really important because, basically the workers are giving them, protein foods, some sugar foods, they're giving them warmth, pheromones, all these different things that workers are providing to the drones to help them reach sexual maturity. Now, what is sexual maturity? So we think about just the anatomy of the drone's reproductive tissues. They have a testes. This is where the sperm is actually produced. And then they have this organ right here called the seminal vesicle. Now, in order for a drone to reach sexual maturity, the sperm has to physically swim from the testes to the seminal vesicles. And what the workers are doing are basically helping the drones reach sexual maturity and helping that sperm physically migrate from one organ to the next. Now, this is important because let's say, for example, a drone produces 8 million sperm cells during development, but only 4 million sperm cell actually, move to seminal vesicles. That means even though they invested all these resources into 8 million sperm cells, they actually only have 4 million available for mating. And so that's why the stage is really important. And this is sort of a picture of what an immature versus mature drone looks like. And so if your colonies are beginning to produce drones, and I'm sure they are now, depending on where you are located in the Western United States, just take the drone and pop them. So squeeze their thoraxes, and what will happen is this organ called the endophallus will pop up. And if you see a brown sperm at the tip of the endophallus, this is a mature drone. If it's sort of a clear structure like this is an immature drone. And what you'll see is if you catch drones that are coming back from the colony, most of them will be mature because they're going on mating flights. But if you catch drones that are sort of in the middle of the brood nest, near the brood frames, these are the type of drones you'll typically see. And so this is one metric we look for when we're measuring sexual maturity. Now, once drones reach sexual maturity and pass what I like to call the teenage stage, the workers are like, we invested so much energy into you, so much resource into you. Now you're on your own. We have other things to do, other drones to take care of, other colony members to take care of, and so now the drones are on their own. And so basically, what the workers are doing, they're kicking the drones to the outskirts of the colony where the honey is stored. So whether that's in the brood boxes or whether that's into the honey supers. But they're doing that because essentially what the drones are doing is they're switching from a high protein diet, which is necessary for them to reach sexual maturity, to a high sugary diet that allows them to go on these multiple mating flights. And so once a drone reaches sexual maturity, they go on these multiple mating flights to these areas known as drone congregation areas. And these congregation areas are sort of these areas where drones congregate to. And we don't quite know why drones choose those areas or what visual or even pheromonal cues they use to find these areas. But they go to these areas somehow and they basically congregate there to really wait for a queen to arrive. Now, once a queen arrives, they'll form these massive drone comets and chase after the queen. And at a given time, there could be thousands of drones at a time, yet a queen is maybe mating with, maybe even 7 to 10 drones during these mating flights. And so a lot of these drones are unsuccessful. And so you can imagine if there's thousands competing for one queen, how competitive this environment actually is. And I'll go to some videos and some data later on, but I just kind of want to give you a quick summary of this mating behavior. And so what actually causes a poor drone? And so like I mentioned, I had these three major points. One is sperm quality or sperm production, which is really determined during development, their ability to reach sexual maturity during the early adult stages, and then finally, their mating behavior once they reach sexual maturity. And so in order for a drone to be successful and really help a queen be successful, it's really determined by these three major points. And so what actually causes a poor drone? One are these environmental factors. And so, these are things that are either controlled by the beekeeper or something that's managed by the beekeepers or some things that are out of control by the beekeeper. And so one is poor nutrition. And so let's say a colony is foraging on an area where there's a massive pollen dearth or, beekeepers not providing any protein supplementation. Poor nutrition can have a huge effect on the drones. And I've actually seen that effect where I've measured, colonies during a pollen dearth, and I've seen the sperm quality for drones being really low. And so what's really important is that the nutrition is really, really high quality for the colony to produce high quality drones that produce high quality sperm. Number two are pests, pathogens, and diseases. And so I mentioned a little bit about Varroa, but if your Varroa get out of control and they're feeding on the developing, pre-pupa of the bees, then that can have a huge effect on the drones. We don't have much data behind that, but I'm just speculating. But I'd imagine if the Varroa are feeding on the pre-pupa, then that can really deplete sperm quality and sperm production altogether. We have a lot of pathogens, diseases, and viruses, and a lot of these affect the developmental stages of drones. And so, if we think about all these pathogens and diseases, they can have a massive effect on the drones. Number three are miticides. And so beekeepers use a wide variety of acaricides or miticides for their varroa management. And I just want to say that this is a really great practice and it's really important to control varroa. And so before you decide to eliminate the miticides altogether because it impacts drones, just remember that varroa management is number one. But these miticides do have a huge effect not only on the queens, and I think Julian has done a little research on that, but also on the drones. Now we know that the synthetic acaricides, such as apivar strips and amitraz, or fluvalinate, for example, these can have a huge effect on the drones and can actually drop sperm quality and sperm viability by at least 50%. And so these synthetic acaricides like apivar have a huge effect on the drones. But even to a lesser extent, oxalic acid, formic acid, Apigard, some of these, soft chemicals or organic treatments, they all have some degree of effect on the drones. It's just not as harsh as, let's say Apivar. For pesticides, and so, I did a lot of my PhD research in the middle of corn country in central Indiana. And so in the middle of corn country, they use a lot of insecticides called neonicotinoids. And these neonics have a huge effect on the drones. And then finally, temperature. And so I think Allison McAfee's done some really great work looking at the temperature and the effect of temperature on drones. But we know that temperature has a huge effect on the drones. And I actually remember, yeah, during my PhD, there was that huge heat wave across the entire country, and I know it really hit the Pacific Northwest really hard. And I actually heard beekeepers reporting that they saw drones, dead on the ground with their endophallus popped up because, it appeared that the temperature had been so extreme that the drones were just basically dying in midair and falling to the ground. And so, as climate, changes and temperatures start rising, or even the weather changes a little bit, this potentially could have a huge effect on the drones. But this all to say that all these environmental factors can impact drones one way or another. Number two are genetics. And so, beekeepers use a wide range of genetic stocks or breeds across the United States, whether they're using pole line, whether they're using, the Russians, or whether they're using their own bred stock. These all have differences in genetics. And because the drones are haploid, these genetics can be even, I guess, more visible with drones versus even queens or other colony traits. And so one way I like to sort of visualize, genetics to beekeepers is let's say we sample 100 random colonies from different genetic stocks. Some of these stocks will have gene C, for example, and if these genetic stocks have gene C, these colonies are likely to produce drones that produce low quality sperm. But some of these genetic stocks may have, let's say, gene B or gene A. If they have gene A, these stocks are likely to produce drones with higher quality sperm. And so, even though there's a massive environmental component, there is a huge genetic component, which I'll sort of cover because I'm really interested in the genetics of this trait. Because if we understand, this gene A, for example, we can help beekeepers breed and select for colonies that produce better drones, through like a genetic test, for example. And so one thing I'm really fascinated in is the genetic factors that cause poor versus high quality drones. And so, I'll really first talk about sperm quality, which is point number one. And so just kind of a quick summary, right? Honeybees go through complete metamorphosis, going from a larval stage all the way to a pupa stage. So they're basically reorganizing all their tissues to go from a larva to a pupa stage. But sperm is only produced during these 2 to 3 days of development, from the fifth instar larva, when larvae are really big and almost overflowing out of the cell, to the prepupa stage. And so sperm is only produced during these 2 to 3 days of development. The sperm tail is formed once the drones reach what we like to call a purple-eyed stage. Now, once they emerge as adults, they can never produce sperm ever again. And so it's largely determine what's happening during development. And so, really, if anything happens during the stage, this can cause, infertile drones or even drones that produce low quality sperm. And so, and like I mentioned earlier, this is really unique to honeybees because even other bee species, and it's pollinator week, right? So even like bumblebees and sweat bees and some of these other bees, these males can produce sperm, throughout their lifespan. But for honeybees, it's really unique and they're only restricted during this 2 to 3 day window. And so one of my major questions was, how much does sperm vary among these different genetic stocks or breeds that beekeepers purchase? And so one thing I did is what I like to call a common garden experiment. And so, sort of as a geneticist, I really hate the environment, makes things really messy and complicated. And so one way, researchers and myself get around this is we raise these different genetic stocks that have differences in genetics, and we raise them in one central environment. And that allows us to control for as much environmental variables as we can. So for example, if we're raising these, 4 different lines in central Indiana, we know that they're all foraging on the same landscape. We know that the weather's the same, the pesticide exposure is the same, and we can control the management among these. And so it's really not perfect, but it's a really great way to control the environment to the best of our ability. And so what I did was I have 4 lines listed here, but I started with 10 different lines. And I chose these lines based on, first, are they part of a breeding program? And so these would include the Purdue mite biters breeding program at Purdue. We're breeding for bees to actually, find mites and chew their legs off. We chose them, like pole line, for example, we're breeding for not only rural sensitive hygienic behavior, but also the ability to grow and be really great pollinators. And this also includes a Russian breeding program. And so we wanted to include these because they're part of a highly selected stocks. Second, I chose these lines based on, what beekeepers are commonly purchasing. And so, a lot of beekeepers are purchasing queens in either East Texas or California. And so I wanted to make sure I represented those distinct lines. And then finally, I chose them based on, how much are the genetics different, right? Because especially in Southern Texas or East Texas, we have a higher proportion of bees that have more Africanized genetics. And so we want to make sure we represented the bees that may have had more Africanized genetics, but also maybe some bees that have, more of the European lineages. And so we want to try to represent, the genetics of the United States as much as possible. And so we started with 10. And during our preliminary research, we found that these 4 lines, and I'll call them line 1, 2, 3, and 4, show the biggest differences in drone quality. And so, for example, some of these lines produce really great drones, and some of these lines produce really poor drones. And so, I'm really going to focus on these 4 different lines, but that's just to say that I worked with about 10 to begin with. And just for now, just full disclosure, I'm just gonna call them line 1, 2, 3, and 4. I'm happy to give you some data on that, but because this is still really preliminary, and I really don't want to call out any specific producer or breeding program, I'm just gonna call them line 1, 2, 3, and 4. But I'm happy to give you a little bit more data at the end, but I really don't want to call out any specific producer or breeding program. And so what we did was we had about 3 colonies per line. And what we did was we raised drones in these colonies and then put them into basically 4 frame nooks with workers from lines that weren't part of this program. So basically the workers that were taking care of these new drones were from different genetic stocks. Because you want to make sure because there could be a worker effect on the drones also. And then basically what we did was we sampled drones at 3 different ages. We sampled them by age 10, 14, and 20. And one reason we did that is because the sperm quality for drones go under what we like to call reproductive senescence, where sperm quality is really low at a young age. It maximizes once drones reach about 14 days post-emergence and then decreases due to natural aging. And really all bee species and all taxa, even humans, they go through this sort of curve where sperm quality is really low at one age, maximized at a certain age, and then decreases over time. And so for drones, it's really 10, 14, and 20. And so we collected about, 100 drones per colony at age 10, 14, 20. And then we measured about 25 different sperm traits. And so this includes how fast the sperm was swimming, how long the sperm tails were, how viable the sperm was, how much sperm the drones were producing. Because we really don't know what sperm traits are actually important, right? Because nobody's ever done that research. So I wanted to measure as many traits as possible to really pinpoint which traits are important. But I'm really going to focus on sperm concentration because what I found is the amount of sperm that drones produce is a really great proxy for sperm quality. And it's really highly correlated to how viable that sperm was. And so, for example, the more sperm the drone produces, the more viable that sperm is. So, for example, if drones are producing 20 million sperm cells, that sperm is about 90% alive. But they're producing, let's say, 1 million sperm cells, that sperm is probably, 20% alive, right? And so it's a really great proxy for sperm viability and other sperm quality traits. And so here are 4 different lines. Line 1, 2, 3, and 4. These are the sperm number. And so on average, drones produce somewhere between 6 million sperm cells. But we actually had some drones produce, 20 million sperm cells. So there's a lot of variation. But most drones are producing somewhere between 4 to 6 million sperm cells. And so here are different ages, 10, 14, and 20. And I want you to focus on line 1 and 4 because these two lines show the biggest differences. And so first, we see the senescence for line 1 and 4, where sperm quality is really low at 10, maximizes at 14, and then decreases at age 20. And so we saw the same senescence. But what's really interesting is drones from line 1 at age 14 produce 6 million sperm cells compared to only 4 million sperm cells for line 14. And this is really interesting because these drones are treated exactly the same. They're raised in the same colony, the same environment, managed exactly the same time of the year. And so we try to control the environment as much as we could. Yet these drones are producing 2 to 3 times more sperm at age 14 than line 4. And so what this shows to me is that it's not just environmental, but the genetics, the genetic differences between line 1 and 4 are really driving some of these differences. And to test that, I measure what I like to call a heritability score. And a heritability score ranges from 0 all the way to 1. If the heritability has a 1, that means genetics are playing pretty much the only role on the trait, and the environment's playing a very small role in the trait. If the heritability is 0, then that means that environmental variation is playing a huge role in the trait, whereas genetics isn't really doing much. Now, we found the heritability for this trait 33%. Now, while this seems really low, this is actually a pretty high heritability, especially for a sperm trait like this. And so if we look at, other livestock or even other bee species, the sperm heritability is usually around, zero point 0.2. And so zero point 0.3 is extremely high. And this shows that, if you are buying a different genetic stocks, these genetics have a huge impact on some of these drone quality traits. But for a breeder, this high heritability is also great because it really allows a breeder to breed for colonies that produce better drones. And so this heritability can be really important for a beekeeper, especially if you want to integrate drone breeding into your program. And so I really want to stop there. I want to focus on, okay, we have the traits and we know that they're heritable, so the genetics are doing something. But next we want to find, okay, what specific genes are causing line 1 to produce really great drones versus line 4 to produce, lower quality drones. And so I'll really spare you on the methods, but what we did was we pulled up the whole reproductive tissue. So that includes the testes, the seminal vesicles, the mucus gland, and then the rest of the reproductive tract. We've pulled that out from 500 drones, and we sequenced those to find out, okay, which genes are causing high versus low quality drones. And we found that there's about 1,200 genes that seem to be involved with sperm production for drones. Now, there's 1,200 genes, but they all don't play a huge role in the trait. They usually fall into what we would call a cluster. And so most of these genes are usually in the outskirts. They probably have such a small role in the trait, not too much, but there's usually one right in the middle that's regulating the trait and has a huge impact on the trait. And this is what's called in orange. And so this is a really important gene. So we pulled this one out to see, what the heck is it doing? And is it playing a really important role on the drone sperm production? And so we pulled out that gene and we found that first, drones are producing higher amounts of sperm in the blue, has significantly higher expression of this gene. And so it seems to play a really important role in the trait. And then we ID'd this gene and we found it's called dynein. Now, I really want to spare you on the jargon because it's really not important. But, one thing to think about a sperm is sort of structured like a boat where the head is the main boat structure, the dynein is the motor, and the tail is the actual rudder. And so when you think about how does a boat move and what's a really important role on a sperm or a boat to move, it's really the motor, because if the motor isn't working properly, then the boat's not able to move and you'll be stuck hand rowing it yourself. What we found is that the drones that were producing less sperm, this motor just doesn't seem to be working properly. Now, that could be due to maybe a lack of spark plugs or lack of gas or some part is broken, but something with that motor on the sperm isn't working properly. And so, you have to remember the word dynein, but just remember that this is the sperm motor protein. So it's extremely important for the sperm and sperm production for males. And so next we'll go into point number 2, and this is sexual maturity. But I want you to remember that sperm motor because that'll tie really nicely into sexual maturity. And so once drones emerge as adults, they spend about 10 to 14 days as adults in the middle of the brood box being taken care of by the young nurse bees. And they do this because the nurse bees are helping these drones reach sexual maturity. Now, like I mentioned in some of the earlier slides, in order for a drone to reach sexual maturity, the sperm has to physically swim from the testes to the seminal vesicles. Now, based on sort of the early results, we found that the motor isn't working properly. Now, how do you think that could affect the drone sexual maturity? Right, right. So if the motor isn't working properly, that sperm might not be able to swim from the testes to some of the vesicles. So one reason that these drones might not be able to produce as much sperm is because this sperm is just not able to swim from the testes to some of the vesicles. So they could still be producing the same amount of sperm during development, but this sperm isn't able to swim to some of the vesicles, then they're really not producing that amount of sperm. So, for example, let's say these drones are producing 8 million sperm cells during development, but only 4 million sperm cells can actually swim. That's all the sperm they can actually use during mating, and that's really the most important metric of sperm quality. And so, one thing I'm really interested in is, from the previous slide, okay, the motor isn't working properly. Is that affecting these drones' ability to actually reach sexual maturity? And is a lower proportion of the drones from line four reaching sexual maturity because the motor isn't working properly? And so basically, we repeated the studies. We had line one through four. So these were the exact lines that we used in the previous study. Remember, line one produced drones with the highest amounts of sperm, whereas line four produced drones with the lowest amount of sperm. We raised them the same um, common garden environment to control the environment to best our ability. And then basically, we took drones from each colony, raised them into a similar colony, so they're raised by the same workers. And then once again, we sampled drones at age 10, 14, and 20, so we can capture this entire senescence. And then basically what we did was we popped drones at 10, 14, 20, and we marked were they mature or were they not mature. And the way we did that was popping the drones and basically seeing did they have sperm at the tip of their endothallus, because if they did, they're mature. If they didn't, they're immature. And so that's how we measured sexual maturity. But just for the sake of time, I'm only going to focus on age 20 because, one of our expectations was, really at age 10, most of the drones shouldn't be sexually mature at this age, right? Because, the sperm quality is still increasing, that sperm still be migrating. And so most of the drones probably aren't sexually mature by age 10. Most of the drones are likely to reach sexual maturity by age 14 when the drones reach their maximum reproductive quality. And then really by age 20, most of the drones should be sexually mature at this stage because if they're not, they may never reach sexual maturity. And these drones may just be effective duds or, even sterile. And so I'm really going to focus on age 20 because most of the drones should be sexually mature at this stage. And if they're not, they may be what I like to call a dud drone. And so these are same lines, 1, 2, 3, and 4. And as you can see, for line 1 at age 20, 75% of these drones were sexually mature. For line 2 and 3, it was around 50%. And for line 4, it was only 35%. Now, there could be method issues, right? Because, only 75% of these drones were sexually mature. But remember, these drones were basically raised in the exact same environment, the same time of the year. Everything was treated basically exactly the same. Yet these drones, most of them were sexually mature, whereas for line four, most of these were not sexually mature. And one concerning thing I just take away from here is, 70% of these drones were immature. And so are these drones duds? Can they ever reach sex maturity? And if they do mate with the queen, how does that actually affect the queen? And so I really do have a lot of questions on these drones that don't reach sex maturity because it could be a really concerning issue for beekeepers, especially if you are using line number four or a line that has similar genetics as line number four. And so, I also had a heritability score here, and I found that heritability was around 44%. So, heritability is also really high for sexual maturity. And so, this means that genetics are playing a really important role for this trait. And so, one reason that line four is producing lower quality drones is because, there's probably certain genetics that's causing that. And so, once again, as a researcher, I don't want to stop there. I want to find, okay, what genes are causing these drones to not reach sexual maturity. And so, I did pull out the genes for the reproductive tract, but I also pulled out the genes in the brain. And what was interesting is I found that, one that sexual maturity is largely driven by gene expression or genes in the brain and not the reproductive tissue. And, I was sort of confused on that right away, but it sort of made sense because in order for a drone to reach sexual maturity and go on these multiple mating flights, they had to go through these behavioral changes in the brain because once they begin these mating flights, they have to change their behavior to, find congregation areas and mate with the queen. And so one thing that might be driving sexual maturity is some of these gene expression changes in the brain. And so once again, I found 825 genes associated with sexual maturity and in the brain. And like I said, most of the genes don't play much of a role. Most of them have a really small impact on the trait, but they're usually in these clusters where there's one in the middle that seems to be regulating the trait and regulating all 825 genes. And so we pull up that one gene to see, what is it doing and how is it impacting sexual maturity. And so we pulled out the gene, and first we saw that genes that were drones that were mature had higher expression of this gene, whereas drones that were immature had lower expression. And so it seems to be regulating the trait. And we found that this, when we annotated it, we found that this gene was called RAS32. Now, once again, I'll spare you on the names because really for you, it's irrelevant. But what I like to call RAS32 is basically the UPS driver, the FedEx driver, the truck driver, some transportation service for the sperm. And so what happens is this truck driver or UPS driver, they start in the brain, and what they do is they pick up all the cargo or everything the sperm needs and basically drives all the way down to the sperm, gives it to the sperm, whether that's the motor, and it actually allows the sperm to move. And so what could be happening here is this UPS driver is, picking up spark plugs or oil or gas, whatever the motor needs, and actually bringing it to the sperm. And so, when we think about not only sperm production, the motor being really important, but for the drone's ability to reach sex maturity, it's really tied to this UPS driver. Now, one question that you probably have is, how does this UPS driver get from the brain all the way down to the reproductive tissues? And so one thing we did was we took our two clusters, right? So we had 1,200 genes in the reproductive tract, and then we had the 800 genes in the brain. And we found that there was, a few genes that were shared between the two, and there's one that seems to be really important. And so this gene could be one reason that truck driver gets all the way down to the sperm. And so we pulled out the gene to see what is it. And we found that it's called Bacadal D, and this is basically the highway for the sperm. So what this does is this highway, it's basically a protein. This protein takes the UPS driver, carries it through the hemolymph or the bloodstream of the bees and transports it all the way down to the sperm to allow it to function. And so if you think about why are these drones not producing a lot of sperm and why are these drones not becoming sexually mature for line number 4, something could be happening at one of these 3 stages, whether it's happening at the UPS or cargo driver stage, maybe something's happening at the highway stage, so road construction, or something's physically happening at the sperm stage. And so, I like to, kind of look at this a bit more, but really the biggest takeaway here is something's happening at one of these 3 stages, and that's really impacting drones for line number 4. But what's really great about this study is we have some key genes, and we can look at this a bit further to see, are these genes have certain variants in line number 4, and maybe that's causing these lines to produce poor quality drones. Okay, so really the first half of the talk, or most of the talk, I really focus on some of these sperm quality traits. But next, I want to focus on mating behavior because it's one thing for the drones to produce high quality sperm, but it's another thing for the drones to find and locate queens at drone congregation areas. And so mating behavior is really important. And so just for the sake of time, I'll kind of summarize this quick because I have some data later on. But once drones reach sex maturity, they go on multiple mating flights to areas known as drone congregation areas. And they basically use these landscape cues or even pheromone cues to find these DCAs. And basically what they do is they wait there until a queen arrives. Once a queen arrives, they form these massive drone comets and chase after the queen. And only a few drones will be successful in mating with the queen. If a drone does mate with the queen, their whole basically reproductive tract will be ripped out and they'll fall dead. So drones can only mate once in their lifespan. And so this is a sort of a picture of a congregation area. And so this is a beekeeper with a stick, and they have sort of a, I think either a virgin queen or probably the queen pheromone. And you see there's a bunch of drones here kind of flying around the stick. And so you can find some really great videos on YouTube. Just type in drone congregation area, and it's a really great video showing this. But there's a really cool study done in Puerto Rico, and they were interested in, not only where are these congregation areas, but how are drones locating these DCAs. And so what they did was they had about 30 to 40 different apiaries marked in these triangles, and they put these small antennas on the back of the drones. And, I wish I could do this. I want to do this eventually, but it's a really hard task because you need like 3 to 4 people in different areas triangulating where the drones are actually going. And so it's a really powerful way to see where the drones are flying, but it's a very difficult task. And so that's why not many researchers have done this. And really in the US, the only research I know that did this was the researcher in Kansas State in the 60s, Loper, I believe, Dr. Loper. And he did this method too and really found some really great DCAs and did some really great DCA studies. But for the study in Puerto Rico, they found that even though it's about 30, oh, that was me. Sorry about that. I think my internet just shut down for some reason. That's weird. Okay. Well, sorry about that. I'm not sure what happened. I'm trying to find it.
We're seeing you, so I think we got internet, but
Yeah, that's really weird. I can't even find my Google Chrome anymore. Here it is. Okay. All this I found it. I'll just open up quick and finish up here. I'm only a few slides away, so
okay.
I've never had that happen before, so that's weird. Okay. Open it up. Okay, share. Okay, perfect. Yeah, so even though there's about 30 to 40 locations, the drones were only going to 4 or 5 different locations or DCAs. And what they also found was that, drones tend to go to multiple DCAs early in their life when they begin their mating flights, but then once they get older, they kind of pinpoint one DCA and go back and forth there. Um, Another cool finding they found was they tend to go to these kind of low valley areas next to sort of an upright structure, whether that's next to a forest edge. Um, actually, a lot of times they're found next to football stadiums because of that, because of the big parking lots next to a stadium. And so basically what I did was instead of using that method, I put these small tags on the back of the drones called RFID tags. And it doesn't tell, it's not a GPS tag, right? So I don't know exactly where the drones are going, but it can tell me when the drone leaves and come back from the colony. And so even though it's not giving me any information on mating behavior, it gives me some really great information on flight behavior. And so I marked about, I don't know, I say about 2,000 drones. So, I think I was dreaming drones by the end of the experiment because I was marking so many drones. But yeah, so I marked about 2,000, maybe about 500 per line. And this is what the data sort of looks like. So for the first 4 days, the drones weren't flying at all. They began flying about 20-minute flights. When they got older, they're flying around 30 to 40 minutes. And then this day was actually raining, so they weren't flying at all. And then the next day they picked up flying again. And so this is some of the data I can get from the drone. Not necessarily know where they're going, but we can get some really cool flight behavior data. And so for the age of first flight, what's interesting, line 1, they began flying around age 8, whereas lines 2, 3, and 4, they began flying around age 6. Now for a drone, 2 days is massive. And so one reason that line 1 drones may produce higher quality sperm is because they're delaying flight by 2 extra days, so receiving care by the workers. And so I'd love to explore this a bit more. For flights per day, line 1, these drones are taking about 10 flights per day, 8 flights per day for line 2, and then 6 for 3 and 4. And so this is a show that there's a lot of variation in some of these flight traits. And so, yeah, so, really the biggest takeaway here is I saw significant variation in sperm quality, sex maturity, and mating behavior. And for some of these traits, I found some really cool genes associated with that. And so what does this mean? I really talked about in the beginning about queen failure. And so one thing I want to do is connect, drone quality to queen failure. And so that's really the next stage of my research. One thing I want to look at is, these drones that aren't reach sex mature at, age 20, are these drones going on mating flights? Are they mating with the queen? And what effect is that happening on the queen? So, for example, if a queen mates with, 20 drones, but 10 are duds, is she going to mate with more drones and make up with that? Can she sense how much sperm she's storing, or is she sensing it based on the number of drones she mates with? We really don't know that. I believe it's probably the sperm, but those are questions I still have. Second is performance may be based on different locations. So that's one reason I really wanted to kind of leave out the lines because I only did this in one common garden environment in central Indiana. So let's say I repeat the study in the Pacific Northwest or California or Texas, I might get opposite results. And so I really need to repeat this in multiple seasons in multiple environments. And then finally, if this is true, right? If their line four are producing, lower quality drones because of the genetics, one thing that could be happening is, when some breeders or beekeepers are selecting, they may be selecting for, one trait, and that's having an inverse relationship on other traits. So, for example, if a beekeeper is selecting for, VSH or honey production, that could be increasing that trait. But for sperm quality, that should actually be decreasing that. And for cattle, we see that exact same trend. They've been selecting for milk production for 60 years and see a huge increase, but the male fertility and cow pregnancy rate is drastically decreasing. And so what we see is when we select from one trait, we typically have a decrease in another trait. And so that could be what's happening. But that's really not to say that we shouldn't be selecting for these rural defense traits such as VSH, because that's really important. One of my goals is how do we simultaneously select for, VSH, for example, and drone fertility. And so, yeah, here's my new email. So feel free to email me any questions. I'm not sure how much time we have, but feel free to email. I'm happy to talk. And just thank you for all the support from all my funding partners. And with that, I'll take any questions if I have some time.
Thank you, Garrett. That was very good. Very informative. Appreciate very much. I had one quick question, and that
Mhm.
is in the teenage age is your point too, the teenagers up to about day 14. You indicated that the sperm must migrate from testes to seminal vesicles. It it is actually a movement that they have to go through. They individual sperm have to do this. There's not sort of like a pushing action or a flowing
Mhm.
action that occurs.
Yeah, that's a really good question. I think it's the sperm swimming, but it could be a mixture between the sperm swimming and a pumping action. We don't quite know that part yet. I think the sperm are swimming, but there could be some sort of pumping action by the queen to or the drone to actually do that. But we haven't really looked too much into that. Yeah, but that's a really great point.
Isn't there then a second point too? Then once
Mhm.
they are then inseminated, they are moved into the sting chamber, the vagina of the
Mhm.
female. Don't they again have to swim?
Well, I actually had this conversation with David Tarpey. So when they're in the oviduct, David Tarpey thinks that the sperm actually swim, but when I actually look in the oviduct, because I've done some studies on this, the sperm tails are so long and such a small chamber that they're so tangled up. I don't think the sperm can actually swim, in the oviduct. Once they get to the spermatheca, then we actually see the sperms sort of separate and they're able to swim. And so what I'm thinking is the sperm actually aren't swimming in the oviduct. I think they're actually being pushed by the queen. And then once they're in the spermatheca, then they're sort of released from all the sperm and are able to swim. But that's some air debate, because I think David Tarpey thinks they actually swim, but I'm not too sure about that.
So actually, it may be in both, some pushing and or some sort
Maybe
of
a
a
mixture of both.
movement and a mixture of
Exactly.
both.
Yep, exactly. Yep. But like I said, this is all, new areas of research. So these are all things that, I really want to answer, but I don't have answers to yet, which is exciting and kind of annoying in the same way.
Yeah, I can see trying to ferret out the different environmental factors during those teenage years, the things that those colonies are subject to during that particular time.
For sure.
is just a landmine. I mean, there could be so many different things that might delay teenagerism or,
Mhm.
the sperm movement. Boy,
Yeah, and with the teenage stage, like Zachary weighing some of his students, probably in 2017, so yeah, almost 10 years ago now. But he found that when he caused the drones to be lower quality by giving them some kind of chemical, right, the workers actually kept them in the colony longer, gave them more vibrational signals, and visited those drones more often than the drones that were higher quality. And so the workers seem to have a way to sense whether the drones are needing more care or whether they need less care. And so I think that's really fascinating, that aspect of it.
yeah, exactly. Uh, let's see. I think I see we have a Q&A question from Jock. It seems that the success of colonies to produce healthy drones could be a parameter to measure colony health. Do you foresee any future testing methods for drones to help predict subtle parameters of colony health that may not be readily visible to the beekeeper?
No, I love that question. I love that question a lot because, okay, when you think about a colony, right? When a colony, to begin with, right, they're focusing not only on growth but also survival. But when they have more resources and the colony is healthier, they tend to to invest more resources into reproduction, which is the drones. And so one thing that we could use as a proxy for beekeepers is if your colonies are producing really great drones and are producing a lot of drones, that could be a really great proxy for colony health because what we see is when colonies are really stressed or not doing very well or have really advanced mite damage, then they tend to just go away from drones because they want to invest more resources into, just surviving. And so, yeah, I'd like to do a bit more research on that, but I really think drones can be a really great proxy for just overall colony health.
Very good, excellent. I did put in the two references that you had cited, yours, the 2021 and the Puerto Rican study. I also put that in as
Y-
well.
Yeah, and if you're a beekeeper, that Puerto Rican study is really, really great. And so, I recommend reading that because I love to repeat that in the United States.
Yeah, I was back in Cornell days when we were working. John Zawolinsky was working on it at that
Mhm.
time. Boy, what an issue. John has, I have heard that drone size affects mating success. Have you looked at drone size? Africanized drones are smaller and faster, he's saying.
Yeah, so I have not looked at drone size. So I did measure drone size. But the problem is, this all comes down to finding DCAs and going there. But one thought I had because I've seen some studies where they've raised drones in worker cells versus drones that were raised just in regular cells. And so the drones that were raised in worker cells are obviously smaller. And they found that, less of those drones were in the DCAs that they found. I'd love to do more studies because I'd imagine that the larger drones would have an advantage over the smaller drones, right? Because you can probably maybe fly faster. I don't know, produce more sperm, but
Yeah,
maybe the smaller drones can fly faster, and maybe there's different strategies in these DCAs. But that's a really great question.
big cars and little cars zipping in and out or versus hogging the
Exactly.
road.
Yep, exactly. And the part for the Africanized drones, yeah, I think there's been studies showing that the Africanized drones are a little bit smaller. The one thing we do know is that if we measure the DCAs in their native ranges, that the DCAs are like different heights, for example. So, I think Africanized drones tend to have DCAs at lower altitudes versus, more European lineages. They leave the colony at different times. So there's definitely differences in the drone behavior between, the bees and their native ranges.
Yeah, exactly. Uh, One question is maybe you've now moved to Texas A&M and it is an extension position. Will you have some time and the facilities, the genetic needs that you have, your
Mhm.
approach at Texas A&M to continue some of this work in the near future?
Yeah, for sure. So we have a really great sequencing center at College Station. So I'd love to do some more of the, the related genetics part of it. But really my goal as sort of an extension specialist is, this is a really great idea, but how is this applicable to the beekeeper? And so really combine the two and be, sort of my next goal. But one thing I'm really interested in, especially in East Texas with all the queen producers, is helping them find DCAs, and maybe they need to, flood areas different ways to get more drones in the DCAs or do different methods like Scott mentioned supplementation to improve drone quality, right? And so I'd love to, kind of develop a genetic test. That's what I'm going to continue on. But the second part of that is also to sort of help beekeepers, better manage these DCAs so they have better mating success and better queen quality altogether.
Yeah, there's a huge concentration there in Big Thicket,
Yep.
East Texas, for a short period. That's going to be part of our topic for our next mini lecture. If I can give the commercial now, that'll be part of our topics as we get to our July mini conference.
Oh, perfect. That'd be great. Yeah, I'd
Yeah.
like to see it.
Earlier this week, Priya Basarati Basu talked about nutrition. And so Scott had a question. What does supplements have on drones? What effect does supplements have on drones?
Yeah, so there's been several studies. Juliana had a really great review on it, and they found that if they supplement colonies with protein, I'm not sure exactly what protein supplement they used, but they did see that it increased drone sperm quality by um probably like 20-30%, but I could be wrong, but it did increase sperm quality and sperm viability. And so
Okay.
so if you're a beekeeper, right, and you're making drone mothers to produce drones, or if you want to, mate during a certain time of year, like I really recommend supplementation, especially if there is a pollen dearth and there's not a wide variety of floral resources.
Okay. Okay. We do have to move on, but Michael, I has a real interesting question. Now, are drones that leave the hive on a mating flight mature? I was collecting drones for it and found 50%
Mhm.
did not have mature sperm. I assume they were not old enough.
Yeah, so what I actually saw that too, especially really young drones, and I wonder if part of those early flights are just helping them reach sexual maturity. Yeah, because that's a really good question. Because I've collected a lot of drones that were coming back, and there is a lot of proportion that aren't sexually mature. But maybe they are duds, right? Because even the drones that were age 20, the ones that marked as duds, those ones were going on consistent mating flights or just flights, but they weren't able to produce sperm. So could be a mixture between, maybe that's helping them reach sexual maturity or, maybe they're just duds. I don't know. Because I get that question all the time from breeders, and I really don't know what those drones are or the age they are.
Yeah, those comets could have a huge number of drones in those
Huge.
DCAs. Yeah.
Yep.
One
And really
last one, then we'll
yeah.
move on. Dom asks, can it happen that drones mate their own queen in flight? Kind of going off another tangent here, but your opinions?
Yeah, it's definitely possible. Yeah, and I've seen, inbred queens before, right? That start laying really spotty brood pattern and start laying diploid drones. And so, it is possible. I think that drones and queens have methods to try to, not mate with a sister, for example, but it definitely can happen, especially if, kind of sort of a queen's leaving a colony, they're mating with a drone in the same yard. That definitely can happen.
Well, appreciate very much your coming on this evening with us. Good luck, best of success with your new position as a extension specialist there at Texas A&M at the university. Getting to know the great groups of beekeepers that are the Texas beekeepers and all those that invade Texas for part of the year as
Yep.
beekeepers. And I think you'll have your hands full. That's a pretty big state. There's a lot of beekeepers, so enjoy that position.
Well, thank you very much, and thank you for having me. So, and maybe we can get Texas as part of the WAS. I don't know, but it's great chatting with you guys.
Okay, we will consider Texas
Okay,
certainly as part of
thank you.
WASP. Okay, thank you, Garrett. Thank you very much. All right, we'll let Garrett go and see about his new offspring there in the family. And we're going to seg now to our second of our presentations tonight. So the EAS mini conference is a committee of three. Myself, and then who you heard from last time, Catherine Thompson, who you heard from our last presentation. And then tonight, our third member of our committee is Monica King, and she has graciously agreed to be able to present some information. Monica is in an unusual place. She is off the grid in a place considerably outside of Tucson. And she's in an area where there are a lot of swarms. The swarms are bees in buildings, and those are are Africanized bees, almost exclusively Africanized bees. And so she's going to come on and talk to us about the adventures of and experiences with beekeeping with these Africanized bees and removal of bees from various structures. Monica, thank you. Thank you for agreeing to be on our program tonight, and welcome.
Hi, thank you guys for having me. And I love your t-shirt, Dewey.
Yeah, did you see that?
Yes, I did. I noticed
Her
that
home
right
club,
off.
yes.
Yes,
I had the
yes.
pleasure of talking last year to the group.
Yeah, so I'm with the Southern Arizona Beekeepers Association, and let's see here if I can figure this out. Where's my PowerPoint? Are you able to see this? Oops, nope. I'm sorry, give me 1 second. Okay. Can you see the PowerPoint?
No, it's not coming up.
Not coming up. Oh, don't do this to me. I had it working this morning with my brother, so now I'm a little frustrated. Let's figure this out. Does anybody have a recommendation on sharing the PowerPoint?
Kurt is our specialist. Kurt, are you?
So if you can go in your Zoom window, if you can go to share screen, there should be a button for sharing the screen in your Zoom window.
Okay.
It's a green button.
Oh, there it is. It's behind. Okay. All right.
And then it'll ask you, it'll go through, you'll see all your windows, all the possible options.
Ah.
How's
Ah.
that? Okay. All right, so I'm a third generation beekeeper specializing in bee removals in Southern Arizona, and I teach quite a few students. And so between myself and my students, we do an average of 225 removals a year. That's the Tucson and surrounding areas. I've gone out to Cerro Vista, all the way out to Sells, and from Maricopa all the way down to the southern border. And in 2008, our USDA lab here in Southern Arizona did some research, and they did feral swarmed and cutout sampling. And they found that from the southern border all the way to the south rim of the Grand Canyon, 98% of everything that they sampled were Africanized. So pretty much when we find a feral colony, we say it's Africanized. We don't do, genetic testing on it. We just assume, especially if we don't see the dot on the thorax, like we recommend to all of our local beekeepers to make sure that they have their European queens. But something I want to point out is that there's quite a few beekeepers that actually believe that Africanized is a behavior and not actually a genetic line. And so I just want to make sure people understand that are watching, because I get a lot of comments. I have a small YouTube channel that I throw up a lot of my removals on, and people just, they go, oh, those aren't Africanized bees that you're working with because they're not defensive enough. And so one of the reasons, and is this, I had it shared a little bit differently earlier, but oh well. One of the reasons that we just basically assume that they are is, again, because of the USDA lab giving us that valuable research. So we see swarms all year long. Some of them aren't the typical swarms that are a split, but actually other swarms like an usurpation swarm. And we also have what I call basically a swarm that's like their last chance at survival swarm kind of thing that the Africanized bees do. But the majority of swarms happen spring and fall. And in my experience, even not established, just as a swarm, 1 in 20 are defensive. And then some people don't realize that, when a homeowner calls us and says that they have a swarm, that many of those so-called swarms are actually open air hives, because that is not uncommon site here in Southern Arizona. And if you are doing Africanized bee removals or catching swarms as a brand new beekeeper, they can get out of hand very quickly. So one big thing is you should have experience with Africanized bees and requeening those bees. So when we're doing the bee removals, or actually the swarms, we try to cage or clip the queen. But always make sure, this is like one of my faux pas that I've had in my removal. I actually have had sun rotted equipment. It happens so quickly here in Southern Arizona. And I was called in on a swarm at the local VA, and the swarm was sitting on the person's truck bed on the wheel well. And I saw the queen immediately. I mean, the minute I looked over the bed, I saw the queen. So I just jumped in the back of the truck with no suit or veil on or anything, just kind of doing my, thing. When I spot a queen, I just jump in and grab her. And I put her right away into one of my queen cages, and it was one of the plastic JZBZ cages, and it had sun rotted. And so as I'm got this large amount of people now watching me, and I'm telling, all the worker bees, here's your queen, come to your queen. And now I've got her in my equipment in the back of my truck, assuming that the bees are all going to come to her. She flew off because she got out. So always check your equipment. That's one of my recommendations. And so, as you can tell in the next slide here, this is the same colony that's established. It looks just like a swarm, and this is I believe in a, not sure what kind of tree, but you can tell once some of the bees were gently vacuumed off, you can see the comb that was left behind. And it's just something that a lot of the people, the homeowners here in Southern Arizona, don't even call us when they see a swarm. So this next one, it was a gigantic colony, was established up in a mesquite tree for over a year in a backyard with children, and they knew about it. And so what looks like a swarm, it actually was a very defensive established colony. So these can be quite dangerous. But then some of our swarms are laying workers, and we're actually seeing that more and more frequently. And one of the reasons I think that is because recently I've caught a swarm just here in my own bee yard, and there was 4 virgins in that swarm, and all 4 virgins were being balled by the worker bees. Every one of them, they were being killed. And so I put them in a box, and I happen to have a queen in my queen bank, and I put her in with them, but I kept the candy plug taped off so that they couldn't chew her out right away. And those bees were laying worker. So then they did accept the queen, and they lived simultaneously until, of course, the laying worker had passed away. So, and the queen took over the colony. But this is just some things that are odd that we see with the Africanized bees. With the cutouts, I always, and the local bee club recommends that a beekeeper has 5 years of experience before working with Africanized bees and the cutouts, because they're just unpredictably defensive. And to start with them in contained, like irrigation boxes, or basically just ways that they just can't get away from you, where we have to chase them down. And this is one thing that just shocks everybody, because I highly recommend, for safety and containment, that you do the removals at night with a red light. And I get a lot of kind of negative feedback about this, saying this is very dangerous for me to be doing this, and that I don't know what I'm doing by doing the removals at night when all the foragers are home and everything. But this isn't for me. This isn't the safety of the beekeeper. This is for the safety of the community and pets and farm animals that might be around these bees. So as you can tell, during daylight hours doing the removal, you can see all the bees are kind of going at my veil. And in the evening, doing it with a red light, as you can tell, I had hardly any flight activity at all. So there's a huge difference. And I wish I would have thought of taking the video on the same, or taking a little clip, a picture of the same colony using a red light and using the white light, so that we can see the difference in the behavioral response that these bees do. There's a lot of beekeepers here in the area that do bee removals during daylight hours. I just find it very unsafe, and I've seen them defend their colony up to 200 yards away. And because of that, I just will not put anybody in the area into that situation. And so, you know, they can kill. I've opened colonies up where under shed floors where there's dead pack rats. And I've gotten owl carcasses out of owl boxes. And so that the Africanized bees have just basically went in and took over these locations. And so, and sometimes when you're disturbing them right away, they're not defensive, but when you get into the brood chamber, all of a sudden they, you know, they turn crazy. And we've had it where our local hobbyists have actually opened their colonies, just assuming that they're Europeans. And they've killed flocks of chickens. They've scarred goat ears, maimed them to the point where they're cauliflowered with scar tissue. And not to mention all the attacks of so-called unknown colonies that get put in the news, like people that have lost their dogs because their neighbor did not know that they had an Africanized colony underneath a shed floor or something like that. So when we're doing our bee removals in Southern Arizona, our safety and containment is priority number one. So one thing that is remarkable with our Africanized bees is that these locations that they choose can be extremely small voids. On the one photo, we have just between two rocks, just a crevice, and the bees made a home right between two boulders. A very small irrigation box, one of the extremely little ones, it doesn't even look like maybe, you know, a couple gallon size as for the void. And inside the saguaros, the saguaro that fell over there had a giant, well, I say giant colony, but it was only like 3 feet by about a foot in diameter inside. And because of this, they tend to swarm very frequently. And it's not uncommon for them to swarm, you know, like 10, 15 times a year. So that's probably why I stay so busy. But as somebody who does bee removals, you want to have always pay attention to what kind of damage you're doing to the homeowner, their property. And so you want to be just extremely careful and do the least amount of structural damage as possible. Of course, this is all done with homeowners' consent. And then you have to be careful of electric lines, because I think there was a beekeeper in Florida that was doing a removal and actually was electrocuted. So if we know that there's electric behind whatever we're working with, just like this colony here that established behind a light bulb, a light fixture, you shut the power off, basically just using some common sense. And of course, like I said, be careful while cutting, because sometimes you don't even know as far as the roofs go or walls go where somebody's electric lines run through. And that's not the only thing that we have to watch out for. Of course, we have black widows, brown recluses, and rattlesnakes. So with this one here, where my smoker is where I was setting up, and I was doing a force abscond and setting up a trap out. But that little brick hole that is to the right of that green bucket is where the rattlesnake in the left photo came out of. And she was sitting there inside that hole for a long period of time while I was working those bees, until I saw bees actually coming out of the hole. So I leaned over and peeked into the hole and saw her coiled up in there. And needless to say, I move a lot faster when there's a rattlesnake than when I'm being chased by bees. So abandoned properties are very common in Southern Arizona. And so with this structure, it's a wooden old adobe house with a wooden floorboards, and the bees were actually on full attack, like on both sides of the house. So we're not sure if it was like how many multiple colonies that we were seeing at this location. We never did do the bee removal here because it's on forest land. And so that one is possibly still there. And then where do you keep your honey? I keep mine in the kitchen cabinets. And so that's where this colony built their home is. This was an abandoned manufactured home on a reservation. A friend of ours asked us to come out and do the bee removal because children were throwing garbage away only 50 yards away from this manufactured home that nobody was using. And the colony was getting so large that the comb was actually hanging underneath the cabinet. They could not see inside the cabinet because, of course, the cabinet door was closed. But my husband and I went out and did this removal, and word got around really fast that we were there, and kids were coming with plates to take the honey home. And I'm pretty sure that around 9 o'clock at night, we were giving kids some good sugar rushes there. But thermal imaging equipment is one of my arsenals that I have in my truck at all times. I keep it on me because, as you can see, the heat signature of the colony, 93, 94 degrees is basically our brood chamber. They don't, of course, the thermal imager won't show us how large the honey areas are, but it at least give us an idea of where to start. So we cut through the drywall, and one of my students is there in the photo just holding a large section of the comb. But again, they just use everything, and nothing is surprising me anymore on what the Africanized bees are using. So overturned garden pots and compost bins are huge favorites, and they literally just fill the voids and swarm just over and over and over again. You can imagine during springtime when there's a nectar flow, how often you will have this requeen. This was the largest open air hive with my husband standing next to it there. That's the largest one I had seen so far. I wish I would have took measurements before cutting that one down. But it was an open air hive that was hidden in a mesquite bosque. And one of our local nurseries was clearing that area to put greenhouses in. And the equipment runner was being attacked by bees. And so they found that. We of course save all the bees when we're doing our removals, except for the drones. I will get to that later. But we rubber band the brood into open frames and try to transfer the brood along with the rest of the colony, and then we reintroduce them back at the apiary. Some of the most unique places, a seed mill, the turbine was completely packed full of bees. And this was just one of the saddest things I've ever done, but the owner wanted it cut open. So we cut open the metal turbine and removed the colony in that 1950s, 1960s seed mill. That kind of just made me sad to see that destroyed like that. And then they choose just anywhere. Like I said, just anywhere is just, there was no hole in this brick wall, but behind a rug that somebody left out when they took it out to clean and hang on the wall to dry, they just left it out a little bit too long, and a colony established behind the rug and just kind of started pushing the rug out with all the comb. And that was an extremely defensive colony. Again, a reason why we do them at night for safety and containment. We do have a number of feral colonies that we know of out in the desert, and I like to check on them because there's an average of about 18 per square mile in southern Arizona, and I like to just see how they're doing on their own in the desert versus in residential areas where there's a lot more forage. The bees do much better in the residential, like if my bees knew how well Tucson, they would all move back to Tucson because there's just so much more diversity in the forage than what we have out here in the desert. I remember my dad once saying that while he was visiting, he's like, it's feast or famine literally in my valley. And so right now we're in a famine again. So They're not always behind the entrances, which I always find remarkable when I'm trying to find them. This colony was actually in the roof. They were going in behind vigas and getting in behind some decorative trim that was on the outside of the window. And so these three on the wall, you can see we cut open the walls just because the bees were traveling through those sections. They actually ate the styrofoam in order to create corridors in those areas in order to get to the roof where they established this ginormous, it was about 2 foot deep by about, I think that was a 3x4 opening. And when we were pulling it open, Zach was pulling the drywall down, just a huge section of honey, just like all stuck together from a previous meltdown. That was refilled had like just dropped right on us, that it weighed about 40 pounds. So it's pretty remarkable what these girls can do. This next removal just shows that I had to find, we used an endoscope, and we found that the bees were running down an electric source, an electric line going into the house. And we had to actually cut through the adobe in order to reach the colony. And that was cut 6 feet away from where the bees were actually entering. These Africanized bees have a tendency to leave their comb. They don't stay with it. One of the big things that I notice is that they, along with the queen, will just take off and go, just head out of, I mean, they'll just abandon the comb completely and just run wherever they can to get away while we're doing the removal, whether that's lifting an irrigation cover off or actually cutting through a wall, and they're feeling the vibration of our like our Sawzall or all of our cutting equipment. They might feel that vibration, but there's some of them that are in full attack mode, but then there's a large group of them that is actually running away from us, especially in the soffits and parapets that we work. And so I don't use smoke around any of those roof situations, because the bees can just basically get so far away from us when we smoke them that they cannot be retrieved. So I tend to just go full force at them with no smoke in those instances. Sometimes I do use a Honeybee Healthy and sugar syrup spritz in the air, not on the colony, but in the air to mask the alarm pheromone. But that's all I'll do. I will not use smoke in areas where they can get deep into someone's house. It's not uncommon for us to find more than one colony in a location. So this was a barn, and this location had one in the roof and 2 on each side of the windows that were established within a week of each other, kind of thing. And so, and like I said, it's not uncommon. I've done 4 separate hives from the underneath of shed floors, even with one of the colonies being right smack in the middle of like a 10 by 12 shed. Like the 4 were one on each corner, one smack in the middle, and then one off just to the side. But the one in the middle was getting their entrance was through a break in the middle of the floor, and they could get out the door of the shed. So just amazing. They're just pretty genius as far as their locations go. We do a lot of trap outs from the block and force absconds from our trees. A lot of the olive trees that we have in the area, mulberry trees, Chinaberry trees, a lot of them have so many multiple openings that a trap out is pretty much not going to work. And so just a force abscond, just making their house as unlivable, undesirable as possible over a period of time will actually make them come out. And so the night when we're working, sometimes we don't get in till midnight, 1 o'clock in the morning. And then there's always cleanup in the next day, sorting comb, of course, checking on the bees, feeding the bees, cleaning up all the sticky equipment, washing our suits and our gloves. It's just, as you can imagine, a mess. I've had to go and go behind beekeepers that were starting out that didn't know what they were doing. So this colony that is like hanging off the soffit there was actually originally in the other side of the home, and it went straight across when somebody did not get the queen and they reestablished. So big thing, you must get the queen. And so if I don't know if you can see it really well in this photo, but the queen is in this soffit area. So I use my iPhone, my camera on my phone a lot to take pictures or video where I cannot see in order to make sure that I got the bees. So if there's anybody that got away from me, or if there's any comb further in or anything like that, we're using our iPhones and our endoscopes a lot. And I have problems many times where my phone, the speakers part on my phone can't hear, and it's because well, it's full of wax. But it's a very valuable tool in doing the bee removals. And this is just a spray foam doesn't work. Bees chew through it, and in this instance, they chewed through, they propolized over, and this one even had a can stuck inside. That's what the red is. One of the homeowners just thought, hey, they can try to get rid of the bees by shoving this can up inside. I believe it was the spray foam can that they were trying to fill the void with, but that didn't stop the bees. So just some more cautions that I found. It doesn't matter how tired you are, you got to keep going. You have to be thorough. If you leave behind too many bees, that makes some grouchy worker bees that get left behind, and that could be dangerous to the homeowner. You have to make sure you have all your equipment with you to do the jobs. And you need to have a location to take these bees. You cannot just take them to someone's backyard for waiting for requeening. You need to have an area. So we have like 14 acres in the middle of nowhere where I bring the bees to do the requeening, and I also bring hobbyist colonies here that have been taken over by Africanized bees, and we bring them out to our location in order to requeen them to take them back to the hobbyist. So that I hate it when people just get frustrated by dealing with defensive bees and quit beekeeping because of that. So I try to assist in any way possible, especially if they have a love for beekeeping. We also have sick and sprayed bees that homeowners will not tell us about. They kind of like will leave that part out, that they were trying to kill them on their own. And so if you've ever met an Africanized hive on full defense after they've been sprayed, they're even worse. These are the kind of girls that do those 50 to 100 stings to your gloves the minute they meet you. And so I've known of some hobbyists that start doing Africanized bee removals that have actually been hospitalized because they got too many stings. And so basically, I guess they say 50 stings or over for people that are not used to being stung are something where they need to get in the hospital. And of course, this is something that has occurred. Even fully suited, even with gloves on, these bees can make it through them. I have a suit. My suit is by Rossmann Apiaries. It's the only suit I will use. It's the fully ventilated master beekeeper suit, and I swear by it, but I still get stung through it. So safety and containment, I guess I can't stress that enough. And we can't kill bees. You have to have an insecticide applicator license, a structural license. So it's even illegal to use soapy water. And so if you're not confident that you can get complete containment, you need to refer it to another company that has the experience and or somebody with an extermination license. This is just something the drones, we're talking about the drones earlier, how the mites really love, multiplying in the drone brood. Well, I do use the drones to pull apart the drone cells in order to look at how many mites that they might have. And this is one of the just wow situations where I found 16 mites of different stages of development in this one drone cell. And I sent bees, worker bees, and drones off to the University of Florida, and this colony tested positive for Africanization. And so the ferrules, a lot of people are saying Africanized bees are mite resistant, and well, not all are. And this colony was not doing that well. And so I sent 6 samples to the University of Florida when they were wanting to do some research on Africanized bees, and all 6 samples that I sent out came back positive. And so if you don't know what you're doing and you're living in a neighborhood and you're taking these Africanized bees that you remove from these unwanted locations and you take them back to your house, the red circle was a hobbyist that was doing bee removals, and the yellow locations were all of the bee removals that I was called in on, that I know of, that were likely bees that got away from these hobbyists. So gifting to neighbors is uncool. So one of the big things is when you do bee removals, Africanized bee removals, you need to know the traits that these Africanized bees come with. One thing is Africanized honeybees, they don't like their queen to be clipped or caged. They just don't. They will leave her. And that's knowing that there wasn't a second, third, or fourth queen in the colony. This is just, they will leave the queen. I will put them through a queen excluder to make sure there's only one queen. And I used to cage them, but now what I do is I trap them between queen excluders. I put a queen excluder between the bottom board and the box and on the top, and just make sure that, and then I just leave them for like 2 weeks until the queen starts laying, they get settled in, and then I'll go in and take out that queen and introduce one of my European queens that I order in from out of state, of course. So they don't like to be clipped either. I had tried clipping wings to hold them down, and that did not work. I had, especially this one colony, that when I removed them, had to have close to 20,000 bees. And by the time she was finished walking out the door and hanging in little bushes, all her worker bees, and the fact that they were not reproducing they just started decline in numbers until she got down to close to 3,000. And I got so tired of it that I just, I replaced her with another Africanized queen from another colony that would stay in the box in order to save those 3,000 bees for the requeening process. But sometimes they just don't want to be confined. They don't want to be held down. They are feral. And so this is just some of the techniques that I use. And then one of the big things that they do is usurpation swarms. And a lot of people from that I have that come to Southern Arizona to become beekeepers after moving from like Montana or even back home. An usurpation swarm is something back home. I would say I'm from Florida originally. We just didn't see usurpation swarms. And so a lot of people don't realize what they are. And so this colony, the box itself, the deep and the medium, are actually Africanized, and they're in the middle of being taken over by another Africanized swarm. And that's just something that they do. So Africanized bees will use up all of their resources. They will brood out, use up everything that they've got in their pantry. And when they're totally empty, there's nothing left for them, and it's in the middle of a dearth, they will actually leave and abscond from their location and go and try to take over another colony. It doesn't matter if they're Africanized that they're trying to take over, or if it's a European colony. It's just survival of the fittest, and who's going to win. Likely the queen that is not laying eggs, and the queen that is more in the swarm mode, her flight physique, as I call it, and she can get that stinger out and fight that the queen that's laying and take over the colony. And this is where our hobbyists in our region get into some trouble, because you don't know when this is happening. You don't notice it. You might, you don't have time to sit there and watch your backyard bees all the time. And so I always tell everybody, check your entrance, use your guard bees before you even open it up, before you smoke your colony in your backyard. Just kind of wave your hand in front of the guard bees and just see what kind of attitude that they're going to have, because we just never know when a colony is going to be taken over by an Africanized swarm. And so basically, here I am holding a frame of Europeans that once were an Africanized colony that I did a removal for. And I think took that, my husband took that photo of me for an article that was in the American Bee Journal that Ron Miksha had done on Dr. Warwick Kerr. So thank you for having me tonight. And does anybody have any questions?
Thank you, Annika. Very definitely different beekeeping for sure. One point of clarification with your sanctuary, when you do a cutout, a removal, and bring it back to your isolated desert location, how long do you let it stay before you then attempt to go in to requeen it with a more gentle European stock?
I like to leave them for 2 weeks.
2 weeks. Okay.
Yeah, minimum of 2 weeks.
And those become your production colonies then? Is that that why one of your uses for them?
Well, I teach so many students, and I actually sell the bees. So to put them back into people's backyards, I need them to be European. So I do keep Africanized bees myself. I use them, I basically call them my donor hives. I do, as long as they're not on that number 10 level of spiciness is what I call it. Like they're so defensive that it's just not pleasant to work. But I find that a lot of my colonies that I really, really enjoy are like a Cordovan Italian crossed with our Africanized drones out here. Those are some of my favorite bees. They're the most productive for me. And sometimes, I wait them out. I do wait them out if they're a large colony. Like if I bring in like a colony that's like 20 to 60,000, I'll just, I just find them remarkable that they were so, they were doing so well on their own. And so those are the bees that I keep Africanized. So I'm not saying I kill all the Africanized queens. I don't.
You do some euthanizing, oh, yeah.
I do not euthanize any colonies. I requeen all of them. I have
Very
not met a colony that I've had to kill.
Okay. You went over insurance earlier, but you didn't say anything about it. So you can purchase insurance to cover yourself
Yes,
for this
can do.
liability.
Yes, you can get liability insurance. It's not very expensive at all. Runs close to $600 a year. So it's for like $1 million or $2 million, I think it is. It's really not expensive at all.
Okay. What about a homeowner if they have a swarm, let's say in their house? Can they get a homeowner's insurance? They have to have a special rider. Do you know that offhand?
No, I've had people, from my understanding, there was a farm bill that has like covered honeybees for the most part. But what people do here is if somebody's got a swarm, it's just basically, I don't think that there's anything that's covered for Africanized bees as far as homeowners go, but the city or county will actually make homeowners do the removals. So there's a lot of homeowners that try to keep the bees in the feral locations. They think that they're living in harmony and they enjoy watching those bees in their gardens. And that's extremely dangerous, but nobody that I know of has ever had a lawsuit over it. Even the dogs that have been killed, the people that have been put into hospitals, like landscapers that the homeowner knew that there was an Africanized colony in their backyard. Landscapers went and started using some weed whackers, and they were attacked, put into the hospital. There was no consequences for them.
Okay. Dom had a series of 3 questions. He says, compared to European bees, how many bees are in a typical Africanized colony?
My averages are probably 3,000 to 5,000. They're not very big,
Okay.
but they do have those, I call them the mothers, and those mothers are those 20 to 60,000 bees in one colony. And those are the amazing queens that I tend to keep.
Yeah, we get them in Bolivia and they have multiple queens when they're that large.
Yes, I found 4 mated queens in one colony, and I wasn't sure, I found the one mated queen, and I had caged, this is back when I was caging them, and all the bees left and went and hung in a tree. And then I went over to the tree and I saw 2 more queens running around on top of the worker bees. So I just grabbed a handful of cages and went back over there, and I ended up caging 4 queens in the one. So it was just an open air hive on a side of a block wall, and I was just dumbfounded. I had never seen that many mated queens in one colony. 2, okay, yeah, 4,
Yeah.
no. So I put the queens in individual cages in different nucleus colony boxes, and I dumped this huge mass of bees all across, all 4 of them. And the next morning, all the bees were trying to squeeze into the one with one queen. And
Yeah.
even though
Yeah.
so it's just amazing watching
It
them,
Yeah,
as they
for
are.
sure.
Yeah.
He's asked a second question. How often do Africanized bee colonies form per year? Do you know?
For the year, from my understandings, it's around 15 to 18 on average.
Okay. Yeah. We get that. It's a mixture between, it's hard to tell. Usurpation and the absconds and
Right.
everything. It's sometimes hard to tell. And finally, he says, does it matter with what type of European honeybee queen you requeen your Africanized bees? Does it matter what type of European queen?
No, they don't like any of them. So if I have a double deep sized colony that I'm requeening, I will split it into 5 small colonies in order to get it to take European queens. I will also, when I kill their queen, I will wait 5 days and then go in and shake off every single frame to look for queen cells before introducing the European queen. And sometimes I even put her in a pushing cage.
Yeah, we wait 2 days, but the same idea, then get rid of them and then requeen. John has a question. How often do you see usurpation cases, like in a given year, percentage basis in Arizona? How often?
How often? The ones that come to me, I probably get about 5 to 10% from my students come back needing to be requeened. It's not a whole lot as far as the amount of bees that we have in Southern Arizona. But to me, there's other mentors in the areas. There's people that choose to euthanize them on their own. So there's a lot of them that I don't know about, but I try to encourage them to just come back to our apiary for requeening.
Kurt said he'd like to weigh in on this as well. Kurt?
That must have been a mistake. Sorry, dude.
Okay, sorry about that. Okay, all right. Jock is asking, what are your best tips for action when a beekeeper notices a usurpation swarm hanging on their colony or next to it, attempting to gain their access to that colony? What's your best action?
Scooping all the bees into a nucleus colony that has a queen excluder in the like one of those rotating disc entrances that has a queen excluder. I like throwing them in there as fast as I can. The colony that is in the picture, by the time I was like, I had just worked this colony, and I was like 6 colonies ahead, and my assistant saw it happening, and he's, Monica, I think we have a swarm landing. And by the time we got over to this one, by the time we closed the one that we were working in, went over to this one, all the worker bees and the queen had already moved inside. I found and neither one were marked because they're both Africanized, and I don't mark the Africanized queens. So I had, of course, the usurpation queen somewhere in there. I had the original queen somewhere in there, and I found one queen on like the second frame, and I found the second queen like on the 6th or 7th frame. So I literally just took both of them, put them in cages, and the bees fed which one that they wanted. So I mean, I could have just let them duke it out, because by this time both of them are in there. And so I don't know which one it ended up being, because unfortunately I didn't have my original queen marked. But basically, if you see it happening, get to that swarm, get them off of it, put in a separate colony, and cage that queen, and not cage the queen, but use an excluder to hold her down. And that tends to stop that. And then you end up with another colony. So
Yeah, it's all in how soon you catch them. I mean, if
right,
they've
it's all in
already in and killed a queen,
Yep.
you don't have much hope of doing anything.
Right.
Alex has a couple very interesting questions. She's asked, how long do requeen colonies last before becoming Africanized again?
Oh, it goes. I've had a Buckfast European colony that lived over 4 years after I switched it from Africanized to Buckfast, and that queen was valid and laying for 4 years, very good quality, and never was taken over by Africanized. And then I've had others. I'm not going to pick on the Cordovan Italians, but they're a little bit more easygoing, I guess I should say, and they're easier for Africanized bees to take over. And so, I mean, it can be as fast as a couple months, and as long as the queen makes it. So, it's really, really hard to say it, and it depends on the area too, and on like if you're in an area that has a large population of established Africanized hives that are fairly large and swarming frequently, then you're going to have more of that possibility of it occurring more frequently.
Yeah, exactly. The other of her questions is a bit of this misconception about what of licensing and permitted uses. So she's asking, I've watched YouTube and Africanized bees getting rid of them using soapy water. How long has it been illegal to use soapy water?
So no, when you're getting paid to do a bee removal, you cannot kill the colony. That has to be done by a licensed exterminator. Beekeepers cannot kill the colony, at least in the state of Arizona. We cannot even use soapy water to kill the colony when we're getting paid to do a removal. But if it's your colony in your backyard, you can kill them yourself with soapy water.
Yeah, I mean, it has to do with licensing, of course, as you
Yes.
indicated, but we're actually using a pesticide at that point, and soapy water has never been registered as an insecticide to kill anything.
Right.
And so that's what it gets into. Yeah, it's
Yeah.
one of those thorny ones.
Yep.
Let's see. Let's see. That the others are saying nice, very interesting. Amazing. Great information. Thank you. And I would again like to thank Monica for serving first on this the mini conference committee of WAS, and for volunteering to be able to tell us about her part of the world where bees are. We've got to agree now, I have to say, just a little bit different, and we have to have some different approaches to them. Thank you so much, Monica. Thank you, Garrett, earlier for the presentation. Next month, about the same week, we're going to have some interesting information on queen and queen rearing. So the July WAS mini conference, I look forward. We'll get information out in a couple weeks on our next one. Thank you all for joining us this evening. Have a good evening. Hope your bees are doing well. Have a good tomorrow, beginning the first day of summer. Good night, all.